Hi, good afternoon. It looks like it's just the four of us for now. We're gonna go ahead and get started. Still not too well, actually, so I'm gonna stay off camera for today. So here is the test two review, right, that I posted a while back, and we're just gonna go through them. First question here: how does the function of DNA polymerase 1 and 3 differ? So DNA polymerase 1 is the main polymerase that is responsible for copying the DNA. DNA polymerase 1 that is responsible for using the template, the parent strand as a template to copy, to build, we'll say, to build a complementary strand. And of course, that can only happen in the 5' to 3' direction, right? Now, DNA polymerase, sorry, that is DNA polymerase 3, I meant. And as for DNA polymerase 1, that will take over as the DNA polymerase 3 approaches an RNA primer. And the job of DNA polymerase 1 is to remove RNA primer and replace with DNA. Okay, so that's the main difference between them. Next one. What is the difference between lagging strand and leading strand of DNA replication? So if we look at the replication fork, for example, okay, let's say we have 5' to 3' here and 3' to 5'. One strand will be synthesized continuously into the fork, while the other strand are going to be synthesized in fragments, right? These Okazaki fragments. And it's going to be moving away from the fork. So the top strand here is what we call the leading strand. Okay, so it's continuous synthesis. And the direction is moving towards the fork, towards the replication fork. Whereas over here on the other strand, we have the lagging strand. Which is going to be synthesized in fragments. Okay. And the direction is like moving away from the fork, right? Both is still 5' to 3'. 5' to 3', 5' to 3'. But it's just, you know, in relation to the replication fork. One is going into the fork, the other is going away from it. And that is that. Okay, next, what do we have next? We have what are telomeres and why are they important? Telomeres are repeats of TTA-GGG found at the ends of linear chromosome. Okay. And the function of telomeres is that they protects the ends of the chromosome. Okay, and delay erosion, or delay the shortening, we can say, of critical of gene. Prevents sounds better. Prevents the shortening of genes near chromosome ends. Okay, because every time you copy your DNA, the DNA strand gets shorter and shorter, right? And so you want to prevent genes that are located near the ends from being deleted due to that process. So having a little bit of telomeres there will achieve that. Okay. If there are 20 chromatids in the cell, how many centromeres are there? 20 would be the answer. So this whole thing is a chromosome, right? And then they're held together at the centromere, and each of these would be a chromatid. Right? So if you have 20 chromatids, right, a pair of chromatids is going to be connected together, right? And that's where you will find the centromere here. Okay, so we have 20 chromatids in the cell, 20 centromeres.
Vincent, is possible to 10 because they have just 20 chromatids, right? So two are paired.
Oh yeah,
So-
sorry, you're right. Yeah, thank you. One chromatid, two chromatids. So every two chromatids is stick together at 1 centromere. So if there's 20 chromatids, you have to divide it by 2. Thank you. So that is the 1 centromere. Okay, so if you have 20 chromatids, you divide by 2, that gives you 10 centromere.
What if it's divided already, like it's not specified?
Yeah, that's a good one, right? Like if it's not divided, like you mean if they're already separated, is that what you mean?
Yes, if
Yeah,
it's
but if
separated
they're
already.
already separated, we won't call them chromatids anymore, right? They would be individual chromosomes on their own, right? I think if it's in any phase and then they are separated, if they get pulled apart like that, right? And they're just moving towards the opposite side, then we would just call them chromosome now. We don't call them chromatids anymore, right? Because sister chromatids are only, when they're adjoined together.
Gotcha. Okay. Thank you.
Yeah, good question. Describe the process of post-transcriptional modification. So after the DNA replication process, we have the exons, and we also have the introns, right? And the introns, they are non-coding sequence, and they will be removed. Okay, so in post-transcriptional modification, we're gonna have a couple of things that are happening. We have DNA splicing. Okay, so we'll remove the introns, and we'll glue together the exons. Okay, that's what DNA splicing is. Removal of introns and joining of exons. And then we also going to do more modifications. Okay, you will take this strand here. Okay, and you will try to increase the stability so that it's not going to break down right away in the cytoplasm. There's going to be a 5' cap. It's made up of guanine. It's called G cap. And you're going to have the poly A tail right here. Okay, on the 3'. That's that. Okay, next one. What is the difference between coding strand and template strand? Coding strand is the one that contains information. To make protein. Okay, so this is the one that you want to be copying. Right. Messenger RNA is identical to coding strand. Okay. Of course, the T becomes the U. Right. So if I give you a coding strand and I ask you to change it to messenger RNA, you would just copy everything, but you change the T to U, right? And the template strand is the strand that will be used, well, as a template to build the mRNA during transcription. Okay. You won't be able to tell which one is coding, which one is template just by looking at it, right? Like the question will have to give you that information. All right, next one. You briefly expose bacteria undergoing DNA replication to radioactively labeled nucleotides. All that means is you will be able to track these nucleotides because of the label. When you centrifuge the DNA isolated from the bacteria, the DNA separates into two classes. One class of labeled DNA includes very large molecules, thousands or even millions of nucleotides long, and the other includes short stretches of DNA, several hundreds to few thousand nucleotides in length. What do these two classes of DNA represent? Basically, the large molecule, that's the leading strand, right? Because it's being synthesized continuously, and then the one with the short stretches, those would be the Okazaki fragments. Okay. So if you didn't know that, now you do. Those are the answers. How are anti-codons on tRNA related to the codes on DNA coding strand? Okay, so like, I'm just gonna summarize this for you again. If you have the coding DNA, right, it's like ATCG, for example, then the template DNA is gonna be TAGC, right? They're complementary, right? And the messenger RNA is going to be identical to the coding, but the T becomes the U, right? And then the anticodon, the tRNA, that's going to be complementary to the messenger RNA. So that would be UAGC. Right, so this shows you the relationship. Essentially, like the messenger RNA is same as the coding, and then the template DNA is same as the anti-codon. Okay. Does anyone have any question? Okay.
Vincent, you said TRAN is the anticodon.
Anticodon, that's right. The anticodon is
okay.
on the tRNA. Yeah, it's at the bottom of the tRNA. Yeah.
Okay, thank you.
Yep. What are the different parts of a gene? We have, we have the promoter. Right? We have the actual coding part, and then we have the terminator. Okay, so this is this whole thing here. Right, it's part of a gene. The beginning end of a gene. So your RNA polymerase, it's going to bind to the promoter. It's going to unzip it, and then it's going to copy the DNA, right? Until it gets to the terminator, and the process will stop. So that's going to make a copy of the gene in the form of RNA. Next part. Suppose the normal protein for a gene has exactly 100 amino acid. Okay, a mutated version of this gene produces a protein that's only 20 amino acid long. Furthermore, the sequence of amino acid is the same for the first 6 amino acid, but completely different after that point. What kind of mutation is this? Well, first of all, the mutated protein is much shorter than the original one. So there has to be some kind of nonsense mutation going on. Okay. But it says furthermore, the sequence of amino acid is the same for the first 6 amino acid only, right? Which means there is likely, it's probably an insertion or deletion, we don't know, mutation, right, of nucleotides, right? Not multiple of 3 though. Okay. Resulting in frame shift. Okay, and then it just happens that there is a stop codon in the new frame. Okay. And that's why it stops after making the 20th amino acid. Okay, so I mean that you can work it all out, right? If it's like the first 6 amino acid that are unchanged, okay, so like the first 18 nucleotides are okay, right? We'll say first 18. Right? So that's why like the 6 amino acids are okay. So your deletion mutation probably happened at like the 19th location, okay, or the 20th. Right? So it's one of those 19, 20, or 21st nucleotide. And one of those got deleted, or there's an insertion in there, and then everything after that is going to be wrong. Okay, and it's only 20 amino acid long. Right? If you multiply this by 3, there is a stop codon at the 61st nucleotide, right? After the insertion or deletion. Okay, that's why the rest of it is never made, right? All right. Compare the process of mitosis and meiosis. Okay, so we did that already. There's actually fill in a chart for it. Unless you guys have specific questions you want to ask for this, I'm not going to go through it again. It's in the study guide, right? There's a table that we did. Make sure you know how many offspring, how many daughter cells we're going to create. Like mitosis, we're going to have 4 genetically identical cells. Meiosis, we're going to, sorry. For mitosis, we're going to have 2 genetically identical cells. For meiosis, we're going to have 4 genetically different, right, haploid cells, right? You should know where the genetic recombination occurs. For example, in prophase 1 of meiosis, there are these crossing overs, right? Everything is summarized in over here with the this table, right? Okay, I already posted the answer to this table anyways, so you should go familiarize yourself with it. Okay, 2 traits exhibit 9:3:1 ratio in F2 generation. Yeah, the last quiz was a little bit tough, right? A lot of people didn't do too well, even though you had 2 chances to do it. So maybe we'll spend a little bit more time on it. How many different phenotypes are possible? Okay, so basically you want to know, this particular Punnett square, right? Okay. And we do this when there are 4 gametes, right? That gives you the 9:3:1, and then we highlight those during our class last time, or not last time, but like when we had this lesson, right? So all the ones that I am highlighting right now, right? Those are gonna be the dominant. If we're talking about 2 traits, right? So all these yellow ones, right? Those are the ones that are gonna have like dominant trait and dominant trait. Okay, we'll say D and D. Okay, so like, let's say big R is round and big Y is yellow. So these are the round and the yellow. And then we're gonna have ones that are dominant and recessive. Dominant for the first trait, recessive for the second one, right? Dominant for the first trait, recessive for the second one. So that would be like round and green, right? And then we're gonna have this one here, which is recessive for the first one and dominant for the second one. Recessive for the first trait, dominant for the second trait, like wrinkle and yellow. And then over here we have recessive and recessive for both traits. Okay, so wrinkle and green. So that creates the 9 out of 16. 3 out of 16, 3 out of 16, and 1 out of 16, right? There are 16 different genotypes, okay, but only 4 phenotypes. Okay. So something like this is useful for a question like this. Okay, let's say, 2 peat plants across. One parent has yellow flower, round seed, a yellow flower and round seed. And the other parents has green flower and wrinkle seed. Right? All F1 individuals have yellow flower and round seed. So this tells you what's the dominant one, right? Okay, like if you breed 2 true breed plants, and the F1 generation is all yellow and brown, it tells you that yellow is dominant over green in this case, and round is dominant over wrinkled. Okay. And then you take two F1 and you cross them and produce 1,000 offspring. Okay. So I can say, I can ask you, here, let me finish the question. Approximately, sorry guys, I'm still all over the place here today. It's like still not feeling too well. So bear with me, please. So approximately how many of the offspring in F2 second generation will be yellow and round? Okay, so like, because you're taking individuals from F1 and you know that these are gonna be heterozygous in all location, which means you know it's gonna be this particular chart, right? The question asks you how many are yellow and round. So yellow is dominant, round is also dominant. So dominant is 9/16. So you're gonna do 9/16 times 1,000 because it says 1,000 offspring, right? So 9,000 divided by 16, 562.5. Okay. They ask you, how many do you expect to be green and round? Okay. Green and round. Well, green is a recessive trait, round is a dominant trait. Recessive and dominant, look, recessive and dominant, that's 3/16. 3/16 times 1,000. Okay, so like it will save you some time on the test. You don't physically have to write out the chart. But I mean, if you feel better, you can have this chart, pre-made, right? And then you just use it as reference. But then, knowing this will also help you save some time. All right. Next, a bird with white feather is crossed with a bird with black feather. All offspring have gray feather. What inheritance pattern can this be? Okay, so gray, right? It's like an intermediate between white and black, right? So this would be an example of incomplete dominance. Okay. If the bird has, white feathers intermingled with black feathers, then that would be codominance in that case. Both shows up, right? There are 20 different alleles that exist for a given gene. How many alleles can a diploid organism have for that gene? Maximum 2, right? Okay. Like if the person diploid means you have 2 sets of chromosomes. So if you're homozygous, then you will have 1 allele. But if you're heterozygous, you will have 2. Doesn't matter. There are 20 out there. You only have 2 sets of chromosomes. You can only have 2. Okay. Next one. Describe the different types of DNA point mutation. So we did quite a bit of that, right? We have, point mutation, which means you change a single one. Right? So we can lead to things like silent, right? Like no change, right? In the nucleotide. It could be missense. Or it could be nonsense, right? Okay, so like you might have to identify them on the test. You're not gonna have to create them because there's just multiple choice. But like for example, UUU becomes UUC. I could ask you like, what is this? right? And you look up UUU, that's PHE. You look up UUC, that's also PHE. So this is silent. There's no change. Missense would be like UUU changing to AUU. Okay, so that's PHE. And then if it's AUU, it's going to be ILE. So that's missense because it's different. Nonsense would be like UGC becoming UGA, right? It becomes a stop codon. Okay, UGC is CYS, and it becomes a stop codon. Right? So your protein is going to be shorter than it otherwise would have been. Okay. What events enable genetic variations to be introduced in the offspring? So a couple of things, right? Crossing over, right? Crossing over during prophase 1 of meiosis, right? That's an exchange of genetic information between the homologous chromosome, right? There is the independent assortment. Right? Remember how the chromosome lines up during metaphase is random, right? So like you might get like the red one on top and then the blue one at the bottom for chromosome 1. And then for chromosome 2, it might be the red one at the bottom and then the blue one at the top, right? Or it could be red on top and blue at the bottom again, but everything is randomly organized, right? Another one here. Right. So like what I'm not showing in the picture is that it's not actually just red and blue anyways. Like the gene has already been shuffled from crossing over, right? So it's a mix of red and blue, but then the orientation of each tetra pair, that's also going to lead to, some randomness as to which gene goes to which gamete, right? Then more genetic variation would be, which sperm fertilizes which egg, right? It's all random event. There's so many sperms. And I mean, like, there's only one egg at a time, but then which egg gets released, right? During that month, for example, that is random event as well. And that will contribute to genetic variation. Okay, next, given the parents of these two genotype, assume simple dominance and independent assortment, what proportion of progeny we expect to have to phenotypically resemble the first parent? Okay, so this is a little bit tricky. First parent is going to be having all dominant trait, all dominant feature. Okay. So let's see. Let's say we do the crossing between. We just look at each gene separately. Okay, so let's focus on big A cross big A, small a first. Okay, so if you do the Punnett square here, you're gonna have big A, big A, big A, big A, big A, small a, big A, small a, right? So even though the genotypes are different, but 100% will express big A. Okay, so as far as the gene A go, everybody is it's gonna look like the first parent. Okay, let's take a look at gene B. Gene B is big B, cross small b, small b. And if we do the Punnett square, big B, cross small b, right? Once again, 100% will be heterozygous and 100% will express big B. So as far as trait B goes, everybody will look like the first parent. Okay. Now there's one more trait to look at. Big C, big C, big C, small c, cross big C, small c. Okay. If you do the Punnett square, you will see there's 75% chance. 75% chance express big C. Right. So phenotypically, right, what proportion of the progeny will resemble the first parent? 100% will have the A, 100% will have the B, 75% will the C. And what you want to do is you want to multiply these together, right? So 100% times 100% times 75% is going to be 75%. Okay. So three quarters of the progeny. Progeny means like offspring, right? Okay. All right, let's take a look at the next one here. Based on the results shown in the table below, determine the genotypes of parent F1 and F2 generation. Okay. So the parent is round cross wrinkle. Round cross wrinkle is gonna give you all round. Okay, and then if you take all the round ones and then you breed that, you take like 2 individuals from F1 and you breed them, F2 is gonna be 5,000 something round and then 1,800 something wrinkled. That works out to be roughly 3 to 1. Okay, so if you see the 3 to 1 ratio, then you know this would have to be pure to begin with. Okay, if you begin with purebred, pure homozygous parents, all the individuals in the F1 is going to be heterozygous, and they all resemble the dominant trait. And then if you take big R, small r, cross big R, small r, right? Like we've done so many times by now. If you do the Punnett square like that, you're gonna get the classic 3 to 1, right? 3 out of 4 will be round, 1 out of 4 will be wrinkle. So, you know, the round ones, right, these are either RR or big R, small r. And then these are the wrinkle ones. Okay. Same logic here. Big Y, small y. Or yellow, big Y, small y. Either homozygous or heterozygous for yellow. And then homozygous always for the recessive trait. Okay, more questions here. In a particular plant, leaf color is controlled by gene locus D. Locus is just the location, right? And they're saying here, plants with at least one allele, big D, have dark green leaves, and plants with homozygous recessive is light green. Okay, so a true breed dark leaf plant, that means big D. True breed means it's homozygous. Is crossed with a light leaf one, so small d. Okay. If you start with true breed, we should know by now 100% of F1 is going to be big D, small d. And then the F1 offspring is allowed to self-pollinate. They cross themselves. Predict the outcome of F2, right? Where, so like this question shows you like a diagram like this. Which of the boxes marked 1 to 4 corresponds to plant with dark leaves? Okay, so like if you do the cross here, right? That's going to be dark leaf, big D, small d, dark leaf, big D, small d, dark leaf. So box 1, box 2, box 3. Right? This just shows you an example of how the multiple choice question is phrased, right? Looks a little bit weird, but it's, you know, it's the same stuff we've been working on so far. When inserting a gene into a plasmid, why must they both be treated with the same restriction enzymes? This creates complementary sticky ends. Okay. If you use different restriction enzymes to cut the gene in the plasmid, then the gene won't stick into the plasmid because the sticky ends will not be compatible. Okay. All right, here is another confusing one. Feather color in bungees, which is a kind of bird, I guess, is determined by two different genes, Y and B, one for pigment on the outside and one for the inside feather. Okay, so the green one are all these, okay, and the blue one are all these, and then yellow are all these, and then white is this. Okay, so like it takes a little bit of time to figure out the pattern, but basically, like if you have big Y and big B, right, at least one big Y, one big B, all this is going to be green. If you have only big B and no big Y, it's going to be blue. Only big Y, but no big B, that's going to be yellow. And then it's going to be white otherwise. So here we have a blue one, okay, a blue one. So how do we get a blue one? Well, blue one means has to be small y, at least one big B, right? We don't know what the other one is. They didn't tell us, okay? But at least one big B. Cross with a white one, which is little y, little b. So what color or colors is or are not possible? So in this case, we have no source of big Y. Therefore, yellow is not possible. And also without big Y, you cannot make green, right? So yellow and green are not possible. Okay, it's possible to make a blue one because we have one big B. It's probably possible to make another white one. Like we don't know what the other one is, right? This one could be a small b, right? Like if we know this is a big B, then you won't get a white one for sure. But like this one, we don't know. It could be a big B, it could be a small b. Okay, but definitely not yellow because there's no source of capital Y from either of the parents, right? And without the big Y, you can't make yellow and you cannot make green. Hope that makes sense. The answer is right here for this one. Make sure you go through this one, okay? I think this is one of the questions in the test where you have to like organize the steps. So just familiarize yourself with this. Okay, let's go through some of these biotech questions. 3 different DNA samples are treated with EcoR1 in separating gel electrophoresis. What is the purpose of the marker laying? This is the marker laying, and the marker laying provides a reference. For the size of DNA fragment. Okay, like if you just see this band, right? Like how do I know how long it is? But if you compare it with the ladder, the markers, you'd be like, oh, okay, it's about 800 base pair long. So this provides a reference, right? These are DNA fragments that are of known length, right? So you know how long they are to begin with. Which sample has the longest DNA strand being treated with EcoR1? The longest one is the one that is closest to the top. Okay, so this is the longest one. Not counting the marker lengths, right? Because the marker is not your sample, right? Like it's just a reference that you purchase, right? And the shortest one, in case you wanted to know, it's going to be right here, shortest. What sample was not cut by restriction enzyme? A, right? Lang A. Sample A. Because there's only one band. Right? So there's probably like no recognition site in that sample. Okay, it was not cut. There are 2 DNA samples with short tandem repeats, polymorphism. Sample A contains CAG repeated 10 times, 10 times 3, total of 30 nucleotides, while sample B contains CAG repeated 15 times, that's 45 nucleotides long. Which sample will be further down the gel when separated using electrophoresis? This is the smaller one, right? Smaller fragment. Travels further. Okay. Next. The plasmid below is treated with EcoR1 and BAMH. When the digested plasmid is separated using gel electrophoresis, how many bands will you see? So EcoR1 cut it once, right here. BAMH cuts it here again. So you will have one fragment, shorter one, and then one longer one. Right? 2 cuts equals 2 fragments, 2 bands. Okay. If this was linear DNA, then 2 cuts going to give you 3 bands, right? You have to make sure you understand the distinction. Gene is inserted into the same plasmid in question 25 using EcoR1. The plasmid is transformed into tube of containing E. coli. You culture all the E. coli on a plate containing tetracycline. What result do you expect? If you insert a gene using EcoR1, the gene's going to go right here. If the bacteria, they accept the plasmid, they will have the tetracycline resistant gene. Okay, so what's going to happen? Well, you would expect some growth. Right? Not everything will grow because not everything will accept the plasmid. Okay, like just because you try to feed it to the bacteria, they're not necessarily going to take it, right? So those that have taken up the plasmid, they will be able to grow in the presence of tetracycline. Okay, everything else would die. List the different types of connective tissue. There's like a whole bunch of them, right? That we that you learned from lecture 10. Okay, so in fact, like we have a chart for something like that, right? So you make sure you're familiar with these kind of connective tissues. Okay, so there's like epithelial tissue. Okay, there's like, you know, loose connective, right? Dense connective. Maybe I'll just show you where to find it because I don't want to relist all of them here. I posted the answer for you in the study guide, but here it is. Okay, like there's a table for it, right? So these are all the epithelial tissues, and then these are the different kind of connective tissues, right? Like there's adipose tissue, cartilage, blood, loose connective, dense connective, bones, right? So you make sure you you know, have this chart filled out, and you can use it as a reference on the test. What type of epithelial tissue is best for surfaces specialized for diffusion? Okay, so there are different kinds of epithelial tissue, right? These are the common kinds. You have the simple squamous, Right? These are flat cells, one single layer. Those are the best one for diffusion. Okay, simple squamous. These are simple cuboidal. They usually are found around ducts. They're good for like secretion absorption, that kind of stuff. We have the simple columnar, right? So again, fill this chart in, make sure that you you have that information. And I already posted the answer to this, so you should be okay. So the answer for this one is a simple squamous. What is the difference between positive feedback and negative feedback? I don't remember if I talked about this in the lecture, but something that's good to know. This is a big thing in second semester. So negative feedback means the trigger of a process is going to result in a series of events that will undo the trigger. I'll give you an easy example. Okay, like if it's hot, okay, if you're feeling hot, what's gonna happen is your body's gonna do a bunch of things. Okay, like it's you're gonna sweat, right? Sweat. You're gonna maybe have your blood vessels dilate a little bit, right, to fan off the heat, right? And some other stuff that we're not gonna talk about now. Okay, but the sweating, the blood dilation, right, that will help you cool off, right? So what was the trigger? The trigger was too hot, and then a bunch of stuff happens, which leads to the opposite. That's called negative feedback. Okay. Similarly, like if it's too cold, right, then we shiver. Right? Maybe you put on a jacket. Well, that's not actually it. Okay, shiver. Okay. We talk about things that your body does, right? Shiver. Your blood vessel bury themselves, buries deeper into the you know into the body, buries deeper, right? Like you might get like goosebumps, right, which causes your hair to stand up, right, in your skin. That traps more heat, and then that would cause you to warm up, right? So again, the end result is opposite to the trigger. This is called negative feedback. Most of the bodily process are gonna have these negative feedback to help you maintain homeostasis, right? Internal balance. Sometimes you will have positive feedback in the body. Okay, so I'll just give you two examples. Positive feedback, like if you have a cut. Okay, if you have cut, you're bleeding. So what happens is you start, so cut, and then it leads to bleeding. And then what happens is you will start to form a clot, right, to stop the bleeding. Once you start to form the clot, you will attract more, we call them platelets. Well, you know, right? Lecture 10, we talk about platelets. And then platelets will form more clot, and then you attract more platelets, and it just keeps on. And then it just keeps on, you know, feeding back onto itself, exaggerating it, right? So that's called positive feedback. Another example is giving birth. Right? There's a little bit of contraction, and then your brain makes more hormone to cause more contraction. And then the contraction keeps on exaggerating until the baby comes out. So those are positive feedback. Right? I guess the main difference is negative feedback means you're gonna undo the change. You're gonna try to undo the initial trigger. Whereas positive feedback, you want to exaggerate it. Okay, you want to emphasize it, amplify it. Okay. This is a very subtle one that it might be like a question on, I don't remember, but it's good to just remember it. An endocrine gland, it's a gland that secretes directly into the blood. Okay. So an example of an endocrine gland would be like your thyroid gland. Okay, it's around the neck, and then it secretes hormones directly into the blood. You have a lot of endocrine gland in the body: thyroid gland, adrenal gland, okay, pancreas. All these are endocrine gland. Exocrine gland, they secrete through a duct. Okay, like sweat gland. Okay, there's a duct that carries whatever they're secreting to a particular place. Okay, the mammillary gland, there's like milk duct in there. The pancreas is both an exocrine gland and an endocrine gland. Okay, so some things has a duct. If it does, we call it an exocrine gland. If it doesn't, call it an endocrine gland. Okay, and if there's a duct, then it's going to be surrounded by those simple cuboidal cells that we were talking about, right? Okay, so that's it. I mean, the test shouldn't be too hard. It's very straightforward. I know the last quiz was a little bit of challenging for some people, but you know, the final test should be pretty straightforward. And that's it. This is our last class. You know, you won't see me again before final test. Of course, you can email me if you have any questions. The test will be available on Wednesday. You know, the usual time from 6 a.m. to midnight, basically. Okay, and then I'll post the marks once everybody have completed that. Maybe I'll see you guys, some of you, next semester. I'm only doing in person for second semester next, in May. Okay, so if you're switching to in person, then I may bump into some of you. Otherwise, best of luck with your summer semester, and good luck with your exams. And thank you for coming all the time to these tutorials. You guys enjoy the rest of your week. Bye for now.
Thank you. Bye.
Thank you.
Thank you. Bye.
Thank you. Thank you.